glad to welcome you guys on our workshop in our molecular biology project on detection of pathogens, and I'm glad to start our workshop devoted to DNA extraction. Today we are going through some theoretic basics about how DNA extraction works, what is DNA and everything about this, and then we'll try to extract DNA and use like the old method, but this method is highlight each important step of any protocol of DNA extraction. To extract DNA, we should destroy not only cell wall, right, but phospholipid membrane as well. If we want to extract DNA from bacteria, and if we want to extract DNA from eukaryotic cells, from plants or animals, we want to destroy one more membrane. But we will talk about that later. Okay, let's discuss what's inside bacterial cell. What we have inside, under the membrane. So, all information about all proteins and structures in the cell contained in DNA. So, we have DNA inside the cell, obviously, we should have. In bacterial cell, we don't have so much chromosomes. They usually have one circle chromosome located in here, in the center of the cell. It's very long, and it's binded to the wall membrane, to the cell wall, yes, to stay here. So this DNA, and DNA contain all information about proteins, about all. How do you call the part of DNA which contains information about one protein? Gene, yes, right, that's right. So DNA contains a lot of genes. For example, do you know how many genes in our DNA? 40. It's close, it's close. This is the number of genes in, right. But in our organism we have about 20,000 genes. 21, about this 20,000 genes. In the typical bacterial cell, for example in E. coli, they have 4,000 of genes. 4,300. Something like this. It's how much genes have E. coli. But it's interesting and strange thing that, for example, we are complex organisms. We are very complex. We can speak, we can think, we can do a lot of things that bacteria cannot do. And it's obvious and it's logical that complex organisms have a lot of genes, right? And simple organisms have smaller amount of genes. But that's not a rule in biology, because bacteria have 4,000, we have 220, but some plants have 100,000 information. Okay, so that's it for bacterial cell. Let's move to more complex situation, because today we cannot extract DNA from bacteria because it's too small and too hard to destroy the cells. They need more complex equipment and they need special kits to purify this DNA. But we can extract DNA from plant cell. And let's remember how plant cell work and structures, right? Yeah, what? Yeah, hemicellulose. Yeah, absolutely right. So if we talk about plant cell, it's hemicellulose. It's a material for papers and for clothes, for a lot of things. Hemicellulose. And hemicellulose, as you know, is a polymer. And what is the monomer of hemicellulose? What is like one block of this polymer? What is it made of? You can eat, I give you a hint, you can eat this. Yes, it's glucose. Yes, it's glucose. Yes, but this glucose combined together by special types of bonds, and these bonds we cannot digest. We, as organisms, we can. So we can eat cellulose. That's why we can eat trees. We cannot eat trees. But some bacteria can, and some fungi can, because they can digest these special types of bonds between glucose. But it is interesting how, like, our sugar, yeah, made some strange things, some strong things as hemicellulose. Okay, we have cell wall, we have cell membrane. Cell membrane contains phospholipids as well, because phospholipids is a common component of any membrane. We have cytoplasm as well, and we have a lot of ribosomes, right? But what we have else in plant cell compared to bacterial cell? Nucleus, yes, sure, because it's eukaryotic cell, we have nucleus. Nucleus is surrounded by membrane, right? What's inside the nucleus? guys. You know. It's in the topic of our seminar. It's DNA, yes, sure. Because why DNA is called DNA? What's N? It's deoxynucleic acid. So we know it's acid, it's located in nucleus. It's deoxy. I will show you later what it does mean. That's not really important, but okay. So here we have DNA. And this DNA contains all genes and so on, as we discussed previously. So, what else? Before we proceed to DNA extraction, well, what else do we have in plant cell? Only nucleus and ribosomes? What process is special for plants, for plant cells? Photosynthesis. So we have chloroplasts. Yes, sure. It's organelle like this with special bodies inside. It's called thylakoids, and they make photosynthesis. Yes, sure. Okay. What else do we have? Mitochondria, sure, because we have to breathe. Yes, and mitochondria is what? Why do we need mitochondria? Tell me. Yeah. In mitochondria occur the last step of cell breathing. Yeah, we have 3 steps of breathing on a cellular level. The first is how do you say glycolysis. When we process molecule of glucose. Then we have cycle of Krebs. You remember? And then we have the 3rd step here in mitochondria. It's called oxygenative phosphorylation, something like that in English, yes. And in here we produce all energy for the cell. And what is the main molecule used for energy in the cell? It's ATP, yes, right. It's ATP, right? That's right, ATP. Yes, and that's the most important molecule because this is like energy equivalent for all processes in the cell. Okay, so we covered all. Oh no, we forget one thing. What else we have inside the cell? Endoplasmatic reticulum. That's right. I will not. It's difficult to draw it because it's a lot of compartments like this inside the cell. But we have endoplasmatic reticulum as well. And what else do we have? From endoplasmatic reticulum, we have some bodies which goes to Golgi complex. You remember? This is the organelle which process proteins after synthesis. Yes. This is very important organelle, Golgi complex. And we have one more thing, it's skeleton, cytoskeleton. The bacteria don't have cytoskeleton, but plant cell and animal cell have cytoskeleton. And it's combined of microtubes. You remember that microtubes are tubes which go through the whole cell, and they need for transportation for it's like roads inside the cell. If you want to transport mitochondria, you transport it through these lines, like rails or something like that. There are special motor proteins which transport big organisms through using these lines. Okay, so we covered the structure. Then we want to extract DNA, right? What do we want to destroy first? This should be here, yeah? We want it outside, in solution. We should destroy cell wall, right? We should destroy cell membrane. And we should destroy membrane of nucleus, right? And moreover, if we want to extract pure DNA, we have to remember that DNA in the nucleus not alone. It's packed with a lot of proteins called histones. You remember? So it's packed like this. This is DNA, and here we have a lot of proteins like histones. And if we will extract DNA, we will extract them as well. That's why a lot of protocols include purification from these proteins, because we want to have pure DNA. These proteins are binded to DNA by electrical interactions. So DNA is acid, right? And in solution, it has negative charge. But these proteins on the surface have positive charge. So they bind to the DNA in special locations, And to get rid of these proteins, we have to change conditions a little bit and replace this. I will tell about this later. So, okay, we know about the DNA, how it's packed, and we know what we want to destroy. Let's proceed to the protocol, how we will do this. First of all, we will destroy cell walls and cells as well. We have strawberry, and this is very important because strawberry is soft. Yes, so it's easy to destroy cells and cell walls, and for this purpose we will use stick. And after that, we have a lot of nucleus in solution, and we have to destroy this membrane. For what we use to destroy membrane and how we do this. You can show on the table what we will use to destroy membrane of nucleus. You can see. You can guess? No, it's not salt. Yes, it's soap. It's soap. It's detergent. So we should destroy nucleus membrane. Why soap destroy nucleus membrane? Because membrane consists of phospholipids, remember, right? And phospholipids in solution makes bilayer. Yeah, this layer of two layers of phospholipids. So phospholipids have two parts. This is hydrophilic part, it's charged, so it likes interact with water. Yes, and this is hydrophobic part, and it's uncharged and it likes to interact with each other. So this is the optimal structure for phospholipids in solution. But soap has different structure. Soap usually has only one tail like this. It's this molecule of soap like this. And in solution, it becomes spherical. It's called micelles. Well, because this is charged, this is uncharged, right? So when we add these micelles to the membranes, these micelles integrate in the membrane, and the membrane can integrate here. So it takes away these phospholipids and grab it to micelles and wash away. That's how we destroy using detergent any membrane, and that's that way we use it in any preparation when we want to destroy cells. Okay, I understand. We destroyed membrane. So now we have DNA in solution, but DNA is still combined with proteins, and we want to as simply as possible, substitute, replace proteins by something else. As we know, proteins bind to the DNA by charge, by electrical interactions. So we have here table salt. We'll add salt, and salt, as you remember, it's natrium chloride, right? And its parts fall into two parts, natrium plus and chloride minus. And natrium ions has the same charge as proteins, and they will displace it. But these natrium ions make a salt of DNA, and this is not brings any impurity. It's not bad for any further preparation. So we can replace these proteins with salt, and it's okay. So it will be more clean DNA. It will not, of course, it will not replace all proteins. Usually in protocols they use special another proteins to cut all proteins and stay and leave DNA. It's called proteinase. Like proteinase K or something like this. It's very aggressive things, like digestive proteins in our stomachs. Okay, so this is the sort, replace proteins. After that, we have clean DNA in solution. We should separate it from solution and leave anything in solution. What we add to separate DNA? Yes, it's alcohol. We can use isopropyl alcohol, but today we will use just regular ethanol because it works as well. And it should be, remember, it should be ice cold. Cold alcohol sediment DNA much better than just warm alcohol. So okay, sediment DNA, but it's not be on the bottom because DNA is lighter. It has lower densities than water in solution, so it goes up, not down. So that's it. That's the principle of DNA extraction, and that's how any preparation works. Somehow, some preparations include additional purification steps, using columns, using spin columns, because they can bind DNA, then we can wash it with different solutions, and wash it from additional salt if we don't need it, wash it from additional proteins if we don't need it, like this, yes? So, but today it's not important. Today we cover the main steps, and we'll see the DNA itself, how it works in solution and out of the solution. the end of any experiment is washing dishes, you know? Don't go away just right like that. So we should wash everything, make the place clean after us, yes. And thank you. I think it was interesting. Yeah.
Okay.
Great. Thank you.
Thank you.